So I know that Morgan posted a few things in the Brain Organoids channel. Maybe we can go through those things and look at them.
Those might have been a while, but yeah, hit me. Or like,
Yeah. So this is this. I remember we talked. I think. Let's see. Is that right? Okay. Yeah.
I put some things from last week on the computational psychiatry.
Okay. Yeah. I don't. Well, we can talk about that separately.
No. Organoids.
Yeah.
100%.
So this is this is Thomas Verbuchen, and this is next generation stem cell models for studying brain development and disease. So he's using this organoid model for looking at this. A few screenshots. What is this from? What talk series?
Oh, okay. This is so yeah. So this particular group, I should say, is what I love about this group is. Sorry, I got the video started. So, Carlo runs a. I think he's associated with the Lieber Institute at Johns Hopkins.
Okay.
And or certainly that's the group that's kind of like the closest to my heart. You know, super focused on developmental disorders like schizophrenia. Really, you know, seeing it as a neurodevelopmental disorder, trying to understand mechanism. Carlo runs a seminar series that is covers all these topics, right?
Yeah.
He in particular. Let me just get his the particular website, right? So yeah, so let me see if I can pronounce his last name right. Tuoni.
Okay.
Tuoni.
All right.
So Carlo Tuoni works on methods and bringing data. So it's coming back to this AI sciences issue, right? Like what do we need to be able to make mechanistic models of neurodevelopment, right? Having large spatial multi-omic data sets is so vital. So let me get his website, because it's like he's, you know, to some degree trying to do what you might say like Russ Poldrack does for neuroimaging data.
Okay.
With Open Neuro. I haven't quite found it yet, but the data, he's got a platform that's called like Nemo. Let's see if I can find it. But you see on his website here, you know, he's particularly interested in these human, you know, cellular variation maps, like how they relate. I mean, but it's so it's not just stem cell variation and embryonic development, but it's like across species, and so one, it's like it's a tool for understanding, right? So the particular talk that you saw there, right, is like, you know, focused on mouse models.
Yeah.
Right? But, right, because that's the one we can manipulate. At least with our current ethics, that's the one we can manipulate. connecting these two humans, right? So there's another guy whose talks I've been watching recently. It's David I don't want to say Reddish. Yeah, right. Yeah, so cross-species translation through computational analysis. Hey, David Rich, I'll find the link. But like this is the point of like trying to some degree do the data wrangling so that you got mouse, you've got zebrafish, you got mice, you got, say, non-human primates, and you've got human data sets, right?
Mm-hmm.
And how to link those, as well as like in each one of these, how do they speak to embryology and its disorders,
Right.
And he in particular his research focus is on some of the tools for data insights in these large multi-omic data sets.
Mm-hmm.
So, I mean, just as an example, so sorry, I'm just finishing up with giving you a link to David Reddish as someone too. You can find like a rich YouTube history, rich publication history on some of these topics. But going back to Carlo, like UMAP, other kind of PCA techniques to, because when you've got multi-omics spatially, and so you're looking at multiple cell types, right? Like how to try and understand these multi-omic data sets and get insights from them in terms of gene expression, relationships, multicellular relationships, multicellular interactions, etc. That's the and he runs a regular series on this.
Okay.
And and then just for it's the Lieber Institute for Brain Development.
Yeah, Johns Hopkins.
This is the I forget, with some of these institutes, like not 100% sure if like is it at Hopkins or is there like work? Yeah, it's near Hopkins, and like Hopkins professors like have like work there but have appointments at Hopkins. They might be private, but the point being that like had a long history of like it has the largest postmortem brain collection of schizophrenia.
Okay. Yeah.
And they've certainly like it's come up before in terms of the computational psychiatry, the brain organoid work that interests me in terms of approaches to try and address questions around schizophrenia.
Yeah. So one of the things he talked about in this talk was this polygenicity of neurological disorders. So this is where they have this GWAS data, and they're looking at the contributing alleles to or loci to the different thing that they're assaying here. So this is from this paper, Trubetskoy et al. In Nature, which I don't have up, but like basically this is the idea he's trying to communicate, that this was, this is schizophrenia, and you have this GWAS, and you have all these different contributing factors. This is not, this is above and beyond, or this is sort of just the genetic contributions. This is not the other contributions. Yeah. So this is, this is just kind of the genetic underpinnings of development. And then brain development is dynamic and complex. This, is, of course, is a slide showing kind of the cortical structure of a 1-month-old versus maybe the cortical structure of an adult. You have this developmental time course from progenitor amplification to deep layer neurogenesis to upper layer neurogenesis, and then to gliogenesis. So you have these stages of human cortical development going from E10.5 to birth. And then you can look at this in mouse, and it would be a little bit different. And any kind of vertebrate, and you'd have differences in how these things proceed in terms of the timing. So this is communicating the point that the GWAS that we saw on the last slide gets sort of put into place in development through this process. And then, of course, if we can understand the process and some of the things that go into brain development at these different stages, and then the GWAS, we have the GWAS information, we can then perhaps understand maybe how schizophrenia develops, and we can understand more about those sorts of disorders. So that's kind of what they're getting at here. And then this slide, which I found interesting, where he lays out, and I think Morgan posted this slide in the Slack, where this is kind of a longitudinal study for embryonic development. So you're looking at pluripotent stem cells here, not an in vivo case, but it's a cell culture thing, where we're looking at these pluripotent stem cells and looking at embryonic development. So you go from this genotype space, which is represented by the GWAS, you have these polygenic contributions. You have the phenotypic space, which is how those things are manifest in the phenotype. And then in the middle, we have this development space, which you can see is kind of represented by these layered surfaces. And so you go from the genotype space, and they get those genes or those alleles or loci get expressed in development in different ways. And then those different ways that they get expressed contribute to the phenotype. And I guess this means adult phenotype. I found this slide intriguing because, for one thing, it's kind of like showing that it's a generative process from the genotype to this developmental representation, this phenotypic representation. On the other hand, it's not quite accurate in the sense that development is sort of a phenotype, and development sets up the adult phenotype, and you have to distinguish between sort of the developmental phenotype and the adult phenotype. On the other hand, you can't really pull those apart too much because the developmental phenotype sort of influences the adult phenotype in different ways. So it's really, this is an intriguing representation, but I'm not, I don't know. I mean, I get the point, but it's a little bit more, the representation maybe is a little bit more complex than this. What was interesting about this is that in our DivaWorm meetings, we've talked a lot about epigenetic landscapes. And epigenetic landscapes were a method or technique kind of proposed by Waddington. And Waddington, when he did this, he just drew these sort of pictures and used this metaphor of a landscape with valleys, and those valleys were called channels or this process of canalization. So you had this landscape where you had these channels that were driven by developmental trajectories. And so you'd start at the top, and he used this ball as the metaphor of unfolding development. And then development would unfold along these trajectories with these branching points. So you would have, and then the ball would represent like a phenotype. And then the ball would move down the channel to a branching point, and then the ball would either go down one trajectory or another, and it would keep doing that until it gets to the bottom of the landscape, which is supposedly the adult phenotype. And the idea is you could draw out these trajectories through this developmental space and define these different phenotypic possibilities at the end. So however that ball moves through these channels, that's sort of the developmental trajectory that leads to the adult phenotype. And one of the things about that landscape was that it was highly metaphoric, so it didn't really have a lot of detail. But actually, he did propose that, of course, this is all underlined by genetic interactions. So if you go back to the GWAS map, you see that there are a lot of contributing loci. They contribute at different amounts. So some are genes of large effect, some are genes of, trivial effect. And then, of course, depending on your developmental state or your developmental period or phase, those genes contribute more or less. So this is kind of, and then he was very cagey. Well, not cagey. Maybe he didn't have the detail in terms of the genetic map. So he just had these kind of these little squares and lines coming up, and it was the idea was basically that was a stand-in for these genetic interactions. And so since that time, people have tried to quantify the map or quantify the epigenetic landscape. And so I think that would kind of be interesting in the context here, which proposes that there's, of course, a genotype that's being expressed. It's expressed into this developmental map, and then that developmental map leads to the phenotype. But it's basically generative in the sense that development sets up the adult phenotype. You can't just have like a developmental process that leads to a phenotype, and there's like one combination of things that leads to that phenotype. There are all sorts of things like things like phenocopies, and you have buffered phenotypes. You also have pleiotropy, so you get different types of phenotypes from essentially the same genes, but the interactions are different, so they lead to different phenotypes. So there are different ways in which this phenotypic space looks that are totally dependent on the developmental interactions. So it's again an interesting set of concepts to play with. Now this other paper that Morgan posted was also interesting. And so that talk is in that talk series that Morgan mentioned. This is Perspectives paper that is primary cilia as architects of the neocortex. So this again is a lot to do with brain development and microcephaly in this case instead of schizophrenia. Primary cilia as architects of the neocortex, roles in brain development, function, and microcephaly. So this is I guess a group out of Denmark and France. And they're looking at primary hereditary microcephaly, or MCPH, a condition closely linked to neocortical development. So initially, MCPH genes were thought to regulate a limited set of cellular processes, but recent studies reveal that many encode multifunctional proteins, often converging on primary cilia organelles that orchestrate the development of the most vertebrate tissues and organs. And so these mechanisms, these organelles that really kind of put together the brain in development. In this perspective article, we examine the role of primary cilia in brain development and explore how disruptions in MCPH and microcephaly associated proteins compromise ciliary dynamics and function. So these cilia are building sort of the tissue. The cells are being put together into this tissue in a certain order, and the cilia are working to sort of build that structure. And then the ciliary dynamics are affected by these genetic states. So the idea is that there's some disruption maybe in the genetics genetic interactions with the genetic states in development and disrupts the way that the brain is developed into this adult phenotype. So they also want to explore how disruptions in MCPH and microcephaly associated proteins compromise ciliary dynamics and function. We highlight additional cilia-related proteins with potential influence on neurodevelopment. By elucidating the connections between primary cilia and neurodevelopment, we aim to provide insights into mechanisms of neuroregeneration. So it also has an important role in neuroregeneration, and they can understand that as well. Ultimately, advancing our understanding of primary cilia may help develop therapeutic strategies to restore neuronal function and improve outcomes for individuals with neurodevelopmental disorders. So this is focusing on microcephaly, which is something that it's a brain disorder defined by reduced brain size while maintaining an overall normal architecture. So it affects the growth of neocortex in terms of brain size. So micro is small and cephaly is head or brain. And then, so the phenotype is manifest by an occipital frontal head circumference smaller than at least 2 or 3 standard deviations below the mean for age, gender, and ethnicity. The diagnosis of microcephaly is usually performed antennaly during routine ultrasonographic examination or postpartum during the neonatal physical examination. Microcephaly can be caused by a number of genetic and environmental factors, including but not limited to in utero viral infections, teratogen exposure, which things like fetal alcohol syndrome, and metabolic conditions of the type listed here. Genetically conditioned primary microcephaly is termed microcephaly primary hereditary, or MCPH. So it's a specific type of microcephaly that we're interested in here, which is typically inherited in an autosomal recessive manner and primarily affects the development of the neocortex. Corticogenesis, the process of neocortical development, unfolds with the emergence of early neural stem cells, which expand in a number of in the ventricular zone of the cortex. So you get stem cells that expand in this ventricular zone. They migrate up and they form adult cells or differentiated cells. And so you get a limited pool of those neural stem cells, and you have less growth of the neocortex. You have a smaller growth of the neocortex, but you have the same architecture. It's just fewer cells. So you have neural stem cells, which give rise to intermediate progenitor cells that initially differentiate into neurons and then later glial cells. Subsequently, these neurons embark on radial migrational and glial fibers, reaching the cortical plate to establish dendrites and axonal connections, and culminating in the formation of the 6-layered cytoarchitecture. And so the proliferation of neural stem cells influences the neuron count, which in turn shapes the size and function of the neocortex. So this is why you have smaller brain size overall. MCPH is genetically heterogeneous, with at least 30 genes listed so far in the online Mendelian Inheritance in Man, or OMIM database, encoding proteins that orchestrate one or more of the patterning processes of the neocortex. In MCPH, gene variants affect primary cilia, which function as cellular antennae that orchestrate developmental processes in most vertebrate cell types. So cilia, they serve as these antennae in the cell. They kind of protrude from the cell, and they have this role of monitoring sort of the developmental process for the cell, so it kind of knows where to go, things like that. So this is a way to orchestrate developmental processes like neural stem cell pool expansion, axon guidance, and neural migration during corticogenesis. So it monitors sort of where the cell is in the brain, where the axon should go. These are all chemical signals, and then pool expansion, I guess, to make sure that there are enough neural stem cells in the neural stem cell pool to know when to stop dividing and having that sort of a self-regulating capability. These findings have contributed markedly to our understanding of brain development and cilia in neurodevelopmental disorders. So in this perspective article, we present an integrated overview of corticogenesis and the molecular genetics of this disorder, with a focus on how selected microencephaly-associated genes function at the level of primary cilium. We examine the multifunctional roles of MCPH genes within a ciliary framework, highlighting those most likely to act directly at the cilium. And then they explore ciliary mechanisms that influence both developmental and regenerative processes, emphasizing the emerging role of primary cilia in neural restoration. So they want to advance their understanding of how MCPH-related pathways shape brain development and may be harnessed to promote neural repair. So they're interested in this disorder, but they also want to know how some of these cilia play other roles in the brain, how these pathways also allow for plasticity, and so on and so forth. So there are a lot of aims in this paper, and they kind of go over what is known in the area. So this is a figure here. This is worth going over. I think this shows neocortical development. So again, you have this time course of cortical development in the human and then in the mouse. So when you see E, it's the mouse, E8, E10, E15, P1 through 3, and then P2 through 4. And then that corresponds with human stages WPC3, WPC5, 10, 22, and 25. So, they have these, basically they've worked out these developmental stages in each organism. And then there's an analogous stage in another organism. So since human and mice are both mammals, you have these stages that are conserved between the two species, and their timing is basically the same, but it's there's a different nomenclature involved. There's variation in the stages in terms of their timing, but they're all basically analogous. So this just shows the different types of cell. You have the cilia on the sort of the edge of the cell. You can see in this epidermal cell. We have motile cilia at the bottom. Sometimes cilia are sensory organs. Sometimes they help the cell move. There are different roles for different types of cilia. You have a primary cilium here coming out of the edge of the cell here. You can see you have different types of cells, neural progenitor cells, neurons, and interneurons. So you see the neural progenitor cell is sort of a precursor to a number of different cell types, and it differentiates accordingly. And of course, the cilia differentiate accordingly as well. You also have astrocytes, oligodendrocyte precursors, and oligodendrocytes. So you have neural precursors, which can become neurons. They can also become astrocytes and other types of cells. You also have these other things like oligodendrocyte precursors that have a more restricted set of fates. This is a radio-migrating neuron and a tangentially migrating neuron. So you have different types of cells emerging at different stages of development. So in this case, we have these NECs that emerge very early, and then you have this ARGC, which emerges a little bit later. There's a pool expansion of these cells. So these are progenitor cells that form, and then they expand their pool size. So as you know, undifferentiated cells or precursor cells divide and then provide sort of constituents for neurons and other types of cells later. So we have this pool expansion of the precursor cells. We have then this differentiation process. We also get migration involving the cilia. So the cilia are basically picking up chemical signals within these different parts of the developing brain, telling the cell where to go, telling the cell where to project its axons, and so forth. And it's moving through the ventricular zone, the subventricular zone, and then into the different layers of cortex. Later on, you get this neuronal organization, and you get tangential migration, so that different cells know how to connect to other differentiated cells. And you see the cilia are marked here to show that they're an integral part of this process. They're sensing their environment, and they're coordinating these kinds of migratory movements and organization into layers and things like that. All right, so that's basically development. This shows the dynamics of primary cilia and ARGC pool expansion. This just shows this primary cilium. It retracts a little bit here. It's basically being resorbed, and it's also then during cell division, it's being resorbed. The cell divides, and then you get these new cilia coming out. So you have ciliogenesis, which occurs in this part of the cell cycle. So you have a G1 to G0, or G1, G0, which is this where you have the primary cilium. You have the S phase where there's this resorption. You have the G2 phase where the cilia disappears. Then you get primary cell division or mitosis. And then you get this ciliogenesis as G1, G0 proceeds. So the cilia are retract and expand as part of the cell cycle. And so that's a kind of an important point when you're looking at these pools of progenitor cells. And these cilia are important for regulating the size of that pool. So if there's a defect in the cilia or a defect in how they are resorbed and generate, then you have reductions in the pool size. Or you could also have expansions of the pool size, and that could be a different disorder. So this is kind of just showing how these are genetically controlled, genetically mediated. So it's not like, we have these, I wouldn't say repeatable results, but we have this sort of tendency towards these things. Then we have in C, sensory primary cilia and radial migration of neurons. So this is the role of cilium in migrating neurons instead of regulating, say, pool size of progenitors. This is when they're moving to their positions in cortex and interacting with each other. So they're actually picking up signals from neighboring cells. They're guiding their axons. They're guiding their position in the cortex, and they're finding a place to settle, basically, and how to interact with their neighboring cells. And so this is where cilium are playing an important role in organizing that architecture. So interestingly, when you have defects in the cilium, you have defects in the pool size of the progenitors, but the architecture remains largely intact. So it's just a smaller cortical layer with essentially the same architecture. So, I don't know, we haven't read the whole paper, but the cilia disorders have maybe a lesser effect on the organization of the architecture than they do on pool expansion dynamics. But yeah, that's basically what they're getting at here in the paper, and they go through a lot more detail in terms of specific loci and different mutations and things like that, and then just talking about how signaling pathways are coordinated by these cilia and how dysfunction can have profound consequences. And so this is a list of MCPH genes and their cellular function. So we know that they're involved. These loci are involved in different cellular processes, and so if we see that there's a mutation in one or more, we know that there's maybe some, maybe they have a function in common, and we can isolate those mutations to those specific functions. Okay, so that's a good paper. It's just kind of thinking about some of the things that are going on in the brain and some of these disorders. And it links back to that talk that Morgan posted before about a more general model of this stuff.
Sorry, I didn't realize my camera was on.
Okay.
Yeah. The really, really fascinating one, right? Because it gets love the infographics on those. I try to point out to people that, like, you know, when I started grad school, you know, it was mostly histology work around schizophrenia.
Yeah.
Right. And it was like post-mortem slices and things like that. And you know, it was very, I don't know the best way to say it. It was very pre-internet, in that, like, there wasn't a lot of digitization. There wasn't a lot of they weren't thinking about biological scale the way that, to some degree, I think we've benefited from the internet scale.
Yeah.
Is that you know, we're obviously talking about one of the most complex artifacts of the universe, and you know, so often in this histological work, they would go in with a theory, which I could sort of get why, but there'd be something like, you know, cell body size or connections or something like that. And this is again getting back to this, like, lack of automation, lack of computer vision / computer intelligence.
Yeah.
This histological work was done manually, right? So, you know, these poor grad students and postdocs that had to do this work, you know, like understanding that this is painstaking, skilled labor.
Yeah.
Right? So to come away with histological analysis that shows that cell bodies of a particular cell type are smaller in schizophrenia versus controls, you know, was like potentially somebody's, like, dissertation work, you know.
Yeah.
Right? And yet at the same time, right? Like, so that would seem remarkable, and it would seem significant. The problem being that what time has shown is that basically anything you choose to go in and look at, there's differences.
Right. Yeah, there are always differences. It's just, what do they mean? You have to interpret them. Yeah.
You know, so one, you don't understand the normative variability.
Right.
And two, it's just like, yeah, there are all these differences, but unless you actually looking for like 500,000 different metrics of connectivity, receptor type, receptor density, cell body size, you know, like every cell has like 1,000 features.
Yeah.
And there are 3,000 cell types.
Mm-hmm.
You know, like, and in any particular area of tissue, like, you know, like there's some variable number of those cell types that are potentially ordered or disordered or connected together or not connected, you know, like so you got this incredible variability across heterogeneity in terms of, you know, the cytoarchitecture.
Yeah.
And you know, so when you see something like that, like it's I mean, that paper, it's such a great testament to the just like and on the one hand, they're kind of focused in on something because they're talking about primary
Right,
cellular,
right.
and but what I love about it is that it exposes the kind of the complexity in which that feature can be, like the many roles it plays, like where it's coming from, what does it influence, all the other factors that come in development, and and um, and it's just the kind of thing where it's just like we need so much more data at such a finer kind of resolution, and like all of it needs like massive super scaled automation because like humans can't take in all this.
Yeah.
And so it's the kind of thing where it's just like this is why I think I love that the Soften team has gone from real-time effort. I mean, part of their commercialization is, yes, you know us from real-time fMRI, which is sexy but kind of useless. But we've got a startup, we've raised money, and we're doing medical reasoning training, and I think their clinical product will be medical reasoning and pathology, right? Pathology as self-supervised computational histology,
Right.
And what I'm trying to get at there is like to link it back is just like, how do you make computer vision not just measuring cell size, but like literally taking in the whole visual input, right? And so like on one hand you want a David Marr, computational building blocks of like you find the simplest geometries for everything, and and you build up something. But like again, like these self-supervised techniques, like their internals are not that, right?
Yeah.
But what you are training, you're training them on tissue slices, right? So, but that being like, we want eyes. We're fine tuning, or we're doing end to end training of eyes for tissue.
Yeah.
Right? Like they look at tissue and they see they can measure anything, and they can both see the complexity as well as take it apart.
Yeah.
And like yeah, like that will be that's what I want to learn from the Soften team, is like how to do that. And I kind of like the fact that they're not 100%. They're not real. They're certainly not brain scientists, right?
Right.
It's just like no. Like let's not. Like our inclination is going to be like to have fully interpretable models at this point.
Yeah.
And like I think you gain from actually following the kind of tech path of make the end-to-end models, do something
Yeah.
and see what they've learned.
Yeah.
What like you'll get some achievements, and then like there'll be interesting things in the wake.